And this is a perfect transition to Stefan Schattgen, who will present on human TFH response to influenza vaccination. And just to briefly introduce Stefan, he is a research scientist and group leader in the lab of Paul Thomas at St. Jude Children's Research Hospital in the US. In 2015, he received his doctorate in immunology and virology at the University of Massachusetts Medical School, where he worked on innate immune sensing of viral infections. And then he turned his focus towards T-cell receptor biology during his postdoc training with Paul from 2015 to 2020. And his current research interests blend computational and wet lab methods for understanding underlying relationships between a T-cell specificity and their phenotype and function during homeostasis, infection, vaccination, and cancer. And I'm very much looking forward to your talk.
Okay, thank you for that nice introduction, Victor. And like I was saying, thank you to the AIR community and organizers for letting us show our work here today. So I'll just jump right in here, and I'm going to set the stage for our main player of the talk today, which is the T follicular helper cell, by kind of giving a background on the biological process that this TFH cell is a main player. And that is in the supporting the germinal center response, which I know a lot of people here work on antibody and somatic hypermutation. And so the TFH cell is a big player within this process. As we all know, immunology and immune responses happen within the secondary lymphoid organs, in particular in lymph nodes, and that's where the TFH cell plays a very important role, particularly within this germinal center structure here. And it's comprised of different areas. So it's microanatomically separated out between T cells and B cells. And it's really only once you get this reaction going that you start getting T cells moving into this germinal center reaction. So in terms of the cell types that we would expect to find within a germinal center, we would see antigen presenting cells like dendritic cells. We'd also see naive or memory B cells, and also short-lived antibody secreting cells kind of hovering around the zone too. And as we initiate this germinal center reaction, what we have is we have activation of these naive T cells and a differentiation into the TFH lineage. And we have our prime B cells entering into this germinal center around the same time. And once these two get together, the TFH cell supports the B cell and its activation, and it goes into this process of proliferation, class switching, somatic micromutation, and it can hang around within this substructure of the lymph node for quite some time before these differentiated and matured B cells actually exit out of the germinal center reaction and circulate in the tissues, form memory populations, enter the bone marrow along with plasma cells. And so really the TFH cells are super important for promoting B cell responses within these germinal center reactions. And in terms of how they do that, the TFH cells can support the B cells both through direct interactions, and that would be through the help of co-stimulatory molecules. So on the TFH side, we have expression of molecules like PD-1, ICOS, CD40 ligand, and CD28. And these interact with their cognitive receptors that are on the B cells, like PD-1 ligand and so on. And through these co-stimulatory signals, it's supporting the B cell proliferation. And another way that the TFH cells are supporting the B cells is through these soluble cytokines that it's secreting into the environment. In particular, one importance is IL-21, which is a strong signal for these B cells to start proliferating within the germinal centers. So these cells are providing direct support to the B cells through cell-cell contact and also through the cytokine secretion within the germinal center reaction. So we were curious as to the role of the TFH cells and also observing how TFH responses mature within humans. Since this really hasn't been too well characterized yet, there's been a lot of study done in mice describing the mechanisms and of TFH differentiation and activation. But within humans, we are still limited to these snapshots. Usually we're taking blood, and we can sample blood from multiple time points. But as I alluded to earlier, the TFH cells are doing their work primarily within these secondary lymphoid organs, and that can be a little tougher to sample, particularly serially over time, to watch their response and how they mature over time. So this work is done in collaboration with Ali Betty, work I'm presenting today. And a few years ago, they performed this study looking at the germinal center responses after a seasonal flu vaccine. So in their study, they had healthy participants who received the quadrivalent seasonal flu vaccine available in the US. And within these individuals, they were serially sampled after receiving their vaccination. We not only took blood, but we also took fine needle aspirate biopsies from the draining lymph node near the site of vaccination of these individuals over the course of multiple time points. And over on the right, you can see how this process is done. So this would be an ultrasound of that lymph node. In particular, here is the germinal center that we're going to be sampling throughout the course of the experiment for this person. This would be like the cortex and the medulla area. And what we can see here is at week 0, this structure itself is quite small, but after 1 week of vaccination, we can already see that the germinal center is starting to grow in its size. And this continues after 2 weeks and 3 weeks, and even it's still quite enlarged 9 weeks after receiving vaccination. And in terms of the responses within these individuals on the study, here I'm showing 3 individuals. We're looking at the germinal center B cells, or the HA positive germinal center B cells, using a B cell probe to identify those cells. We can see in 2 individuals in particular, they had a very nice response in terms of anti-HA B cells. That happens 2 weeks or so after vaccination in this individual. And in this individual, we can see more of a slow climb in that response over time here. So we're excited to use these samples and these data to kind of explore the other side of the equation and look at what the TFH cells are doing. So in terms of what the TFH response is to influenza vaccination and what we know already, what's been described in the literature, we know that there's a transient increase in the frequency of circulating TFH cells in the blood. And the peak in the cTFH frequency occurs about 7 days post-vaccination, and this correlates with an increase in antibody secreting cells and increase in serum titers. We know that the TFH compartment certainly contains specificity for flu antigens. Flu-specific clones have been identified and also have been noted to be shared between the cTFH compartment and tonsillar TFH cells. And we also know that the cTFH clonal lineages are quite long-lived. So we know that there's memory for these antigens that are still in circulation for quite some time. Now, some previous work in our lab, also done in collaboration with Ali Betty's lab, was looking at the response to SARS-CoV-2 vaccination. In particular, we were able to identify one immunodominant epitope in the spike protein of SARS-CoV-2 that we were able to generate a tetramer and track the responses of these after SARS-CoV-2 infection, both in the cTFH and lymph node. And what we could see within this context is we see this, again, transient increase in the cTFH cells. Here it's later than the 7 days of observed in the flu vaccination, probably due to a lot of these individuals being naive to SARS-CoV-2 infection. But what was surprising to us is that the frequency of the germinal center TFH cells, so the lymph node TFH cells from these individuals, were very high, and they remained very high for a very long time. So out past 6 months here, we could see that there's still upwards of 10% of the TFH cells in the lymph node were actually specific for this one particular epitope here. So this led us to ask, what does the TFH response in the lymph node look like after an unadjuvanted seasonal flu vaccination compared to an mRNA vaccination here? And what changes in their gene expression program are associated with productive germinal center responses? So I'll describe the data set that we're going to be working with and showing you today. And to tackle this question, we use multi-omic approach where we use the 10x genomics platform to generate 5' single-cell gene expression and repertoire. Particularly, we also did PCR, but today I'll be talking about TCR repertoire profiling. And we did this from bulk PBMCs, bulk lymph node, fine needle aspirates, or sorted B cell subsets. And here we can see all the individuals that were included in this part of the study, where we have donors 4, 5, 7, 8, where we have 10x data collected from these individuals. And for donor 11, we only have bulk repertoire sequencing from sorted TFH cells. For the focus of my talk today, I'll mostly be talking about donors 4 and 5 here. These are the best and most comprehensively studied, particularly donor 5, where we have lots of time points where we have matching blood and lymph node samples to look at. Whereas with donor 4, we have mostly just lymph node samples. An extension of what was previously done with these study is, in addition to the first year of vaccination, which was previously published, we've also re-vaccinated these individuals and were tracked out for a second year. Here we're just looking at the lymph node biopsies of these individuals during the second year, but it's a really exciting opportunity to kind of look at what happens after this first sort of exposure here, and then vaccinating them 1 year later. And I will note that these subjects had not received a vaccine for at least 3 years prior. Whether or not they had a flu infection asymptomatically is a little harder to say, but we can at least say that they did not receive a vaccine. Okay, so if we take a look using flow cytometry at the TFH response in donor 5 in particular, we can see both within the blood and within the lymph node here, you get this nice increase in TFH populations. So up here in the PBMCs, we're looking at the cTFH population defined in skates right here. And you can see that at day 0, they're quite small. And at day 5, they had expanded out quite significantly. And associated with that, we see increase in activation markers within those cTFH cells. But by day 12, we kind of contracted back down and kind of lose that activation status here. And we can see this visualized on this plot over here on the right, where we get this quick transitory increase in those cTFH before they kind of go back down. On the other hand, the lymph node germinal center TFH cells, we can see here day 0 started quite small. And by day 12, we have a nice increase, and it keeps increasing at the day 60. And you can see this again visualized over here, where we have this increase at day 12. We're starting to fill up that germinal center with the TFH cells, and then they're maintained within that site for quite some time. Even at day 180 after vaccination, they're still at a higher frequency than we found them to start with. And if we look at the germinal center B cell frequencies during this time, we can see that there's this nice correlation between the germinal center B cells and the germinal center TFH cells. So really, demonstrating that those TFH cells within that lymph node are supporting the germinal center B cell proliferation and expansion during that time. Okay, so jumping into the single cell data, here we're looking at all the T cells that we collected in the data set. It's about 150,000 or so. And we're going to cluster all those together and kind of pick out the TFH cells from here so we can further investigate those. So up on the left here, this is a UMAP embedding of that data, showing all the different clusters that we're able to identify. And if we put some labels on those, we can see, and break those down into 3 broad groups here. We have the naive, we have effector memories, and we have some TFH Tregs. And the reason we kind of had to group these together is there's actually a lot of commingling of the Tregs in the TFH cells within cluster 14 here. That was kind of hard to tease it or pull apart at this level. So here we can see some of the markers across this data set. So CD8s and CD4s and naive memory markers here. But the markers we're going to focus on here are those associated with the TFH cells. So in particular, we have a transcription factor COX-2, CXCR5, PD-1, ICOS, and IL-21, which I said is an important cytokine for TFH supporting B cell proliferation. And we also can see FOXP3 down within this cluster too. So in order to capture the TFH and Treg clonal lineages, we first subset on the CD4 positive T cells, and then we select out the TFH and TFH Treg clusters. Then we take those TCR sequences within those cells that we pulled out and then match them to all other clusters to capture any other cells that may have fallen out or have deviations in their transcriptional profiles that might be real biologically relevant. And then subset on those clonal lineages. So in the end, what we have is about 6,000 cells and about 4,000 unique TCR clonotypes that we're looking at. Now, before I go into the gene expression data too much for these, I wanted to just talk about the TCR clonal dynamics of these samples first. So first, we're simply just looking at the frequency of the TFH cells within the lymph node and also the clonality over this time course. And what becomes immediately obvious is that there's this correlation, a linear relationship between the frequency of the TFH cells and their clonality at that time point here, which we can see over here. So basically, as these T cells are increasing in their frequency, it's due to clonal expansion, not due to lots of new clonotypes necessarily coming into the germinal center reaction. But what was really unique about this study is our ability to do this longitudinal tracking of these clonotypes over time, and not only over time, but across tissues as well. So here we're looking at all the TFH clonotypes we pulled out for donor 5. And here on the top, we're looking at the lymph node. And here we're looking at the PBMCs here, and the size of the node will correspond to the number of clones we're observing at that time point and location. And then the width of these edges between these nodes here corresponds to the number of clones that are shared between these two time points. So first, we can look at the blood and early time points within the lymph node. And what we thought was very intriguing right away was that if we look at the blood at day 0 before vaccination, we can see these TFH clonotypes that are also detected within the lymph node of this individual at day 12 after vaccination, suggesting that there is memory for flu within this compartment here that may be entering into that lymph node in response to the vaccine and expanding out. Now, if we look at this early time point where we have the most clones within there and see how well they're tracking out later, we can see that at day 60 and day 90 after vaccination, so really kind of the peak of that germinal center response, a lot of the clones that had entered at day 12 are still present at these time points as well, and kind of just hanging out in there. Now, if we look at a later time point at day 180, we can see that the number of unique clones within the lymph node has shrunk back down. But we can also still see that a lot of the clones from day 180, if you kind of track this line here, will actually come back or hang around within the lymph node, excuse me, prior to revaccination during year 2. And then after receiving that vaccine on the second year, we can see again, a lot of these clones are maintained within the lymph node and actually begin to expand out at day 7. And are maintained for quite some time within the lymph node structure. So really these TFH clonotypes from this network across these different tissues that is quite long-lived and durable. Okay, so that was just my little bit about the TCR repertoires. I'm next going to talk about the gene expression of these TFH cells. So we know that TFH differentiation happens on a continuum. It's not binary by any means. And so depending on where you are within this response, you might have different phenotypes associated with those TFH cells, which makes it a little tricky to study. But I just wanted to kind of set the stage for some of the subsets of the TFH cells that I'm talking about next. So early engagement of those naive T cells will have some differentiation of those T cells into early effectors, and then they further differentiate into pre-TFH cells. And this depends on the microanatomical location of these. So they start getting up to that B cell follicle there and start interacting with some of the antigen prime B cells before entering into the follicle and acquiring that full germinal center TFH phenotype here later on. And then after that resolves, these TFH cells can exit out of the follicle and form memory. And upon rechallenge, you can actually get recall of these memory responses within that particular follicle. So if we look at the gene expression of the TFH cells and the Tregs that were pulled out, we could see that they acquire these, have these distinct phenotypic states that we can observe. Here we included some naive CD4 T cells just as some context to compare the other populations against. And so we can see here this clusters of pre and memory TFH cells, it gets a little hard to tell whether they're coming or going, and generally share sort of similar phenotype. We have our Tregs over here, and then we have our germinal center TFH cells over here. We also were able to find this population of IL-10 TFH cells, which have been recently described in the literature, but it's not really understood what the function of those IL-10 positive TFH cells are within the context of immune responses, just that they've been observed. But we were able to see that these IL-10 TFH cells actually had quite distinct phenotype from the germinal center TFH cells. And if we look at some of the markers associated with those particular ones, we can see lots of activation markers within our germinal center TFH cells here. So, we could see things like CXCL13, which is important for B cell and T cell homing, structure, we see PD1, upregulation, CTLA4, et cetera, et cetera. But particularly, we can see IL-21 is very nicely produced by our germinal center TFH cells and also the IL-10 TFH, but not really within our other populations here. So all in all, we're capturing these discrete states of the TFH response within these lymph nodes. If we take a quick look at some of the enriched pathways in these different populations of TFH cells, what we're able to confirm that previous reports within mice that the germinal center TFH response really is glucose hungry. So we see this switch to glycolytic metabolism within the germinal center TFH cells compared to the naive or pre-memory. And if we look at these in IL-10 TFH, some of the enriched pathways we observe in those, we can see things like response to cytokine stimulus, cytokine-mediated signaling pathway, suggesting that there may be cues within the environment that is switching them into this IL-10 TFH subset here in particular. So next, if we take a look at how the composition of the TFH compartment is changing in the lymph node over time, we can see starting over here at day 0 in the first year that we have this population of pre-memory and germinal center, so everything's pretty calm and quiet. But after vaccination, we get this quick increase in the pre and memory TFH cells as the germinal center TFH cells are also starting to rise. We also see this increase in the frequency of the IL-10 TFH cells during this time. And then as we go out later into kind of the peak of the germinal serum reaction around day 60 here, we can see now that the frequency of the germinal center TFH are higher than those of the pre-memory. IL-10 TFH are still present. And we can also see that the germinal center B cell frequency is increasing during this time as well. And these germinal center TFH cells will be maintained for some time before they kind of go back down and the pre-memory phenotype rises up again here. And before the second year of vaccination, we can see that, again, things had quieted down, similar to what we've seen in the previous year. But what we see now in terms of the kinetics of the germinal center TFH in particular is that we see a quick increase in the germinal center TFH frequency after revaccination during that second year, much quicker than we saw within the first year. And again, corresponding increase in the germinal center B cells during this time before kind of dropping off a little bit. And again, we detect a very high response later at day 120. It's possible that day 60, we just didn't quite hit the germinal center when we did the biopsy on this one. But all in all, we can see that the composition of these TFH cells is changing over the course of the immune response to the vaccine within that lymph node. Okay, so we wanted to next focus on some of the clones, the TFH clones, and try to deorphanize those and also look at how the phenotype of the TFH response can vary within a single clonal lineage in particular. So we chose 12 TCRs to screen and try and deorphanize in terms of their specificity. And those we chose were, we prioritized ones that we detected both in the blood and the lymph node, ones that we detected over multiple time points. So you can see here within these 12 clone lineages, particularly like clone 3 here, we can see that was present in the blood at day 0, but then we detected day 12 after getting the vaccine, and it persists for some time before dropping out. And we can also see it within the second year here too, that we see some other clones also coming back in this time too. So all in all, these clones here seem to be playing, have these active dynamics in the context of the vaccination and are likely recognizing the flu vaccine itself. So we wanted to try and figure that out. So this work done by Mohammed, we clone those TCRs into Jurkat T cells, and then these will be our T cell lines. And then on the other side is our APCs. We use either K562s, where we put a single MHC class 2 allele into those, or we use donor PBMCs that had HLA, or that were HLA matched for our donor that we clone the TCRs from. We would infect them with flu or do flu segment transfection or peptide pools to try and figure out what exactly those TCRs are recognizing. And we've been able to deorphanize 5 of 12 so far. So you can see for TFH1, TFH3, and TFH12, these are all recognizing internal proteins, in particular M2, M1, and MP peptides within these HLA alleles here. 2 others we were able to identify as specific for HA and for NA. Here we use some plasmid truncations to identify which region of HA that this particular TFH11 is recognizing. We can see it's here within this stretch. We get this increase in CD69, interferon gamma positive. And then for our clone 6 here, using peptide pools for each of the flu proteins, overlapping peptide pools, so it's complete coverage. We can see that this clone was specific for NA from this donor here. So for 5 of the 6, we're able to at least figure out they were specific for flu. And now this lets us get to look a little bit more at how these particular clonal lineages are maturing over the course of the response here. So this is ordered by time, going left to right here. And then we use some of the differentially expressed genes for each of these different populations here to define these modules and come to a single score that kind of captures that signature. And what we could see here is that early during the response, we have this incoming memory pre-TFH differentiation corresponding to high pre-memory module scores. Before those sort of drop off later on at day 28, day 60, and day 90, and now we're seeing this mature general center TFH response taking over. We have a later appearance of these IL-10 TFH cells within the reaction. We also see a little bit early on too. And what was really interesting is that compared to these kinetics that we're seeing here within year 1, in year 2, we're seeing that they're re-expanding and re-engagement as germinal center TFH cells is happening much quicker in the second year. It's marked by this module score, high module score for the GC TFH here. And if we look a little closer at a couple of the individual lineages, we can see some interesting patterns occurring here. So in this particular one, TFH clone 1, this one was one that was detected early on and it's likely a memory TFH cell. So we can see that early on, this particular clone is even making CXCL13, CXCL21. And we also see a little bit of the pre-memory markers within there too. But overall, it suggested that this lineage was a little bit more mature than this clone 3 over here. So if you look at clone 3, you can see a lot of these early markers here, like these VIM, TMSD10, and KLF2 are associated with the pre-memory TFH cells, whereas we don't see IL-21 being produced early, but later on within this lineage too. So basically what we're able to figure out is that within a single clonal lineage, each of those TFH states is attainable. And so just to summarize, these unadjuvanted inactivated flu vaccines can stimulate a GC response. There's significant overlap in the repertoires between different tissues. Their phenotypes fall into distinct states and the composition will change over time. The recall and differentiation into GC TFH with faster kinetics during that second year. And we were able to identify TFH cells that are specific for both flu and, internal and surface proteins of flu. And just as a closing thoughts, so one thing we'd like to understand better is, do the qualities of TFH response to vaccination differ depending on the vaccination strategy? So here we're looking at unadjuvanted inactivated. We can compare that to a live attenuated or mRNA. And some things we need to think about are like, what are the implications of antigenic sin of the TFH cells on de novo B cell responses? In particular, we found that these TFH cells are responding to internal proteins of flu. So how does that limit engagement of new B cells against new HA and NA subtypes? And also further understanding the mechanisms regulating TFH transcriptional reprogramming over time. And with that, I'd just like to point out our paper is now on bioRxiv if you want to go check it out there. And I'd like to thank Paul and the lab for supporting this, and also lots of people at St. Jude and our collaborators at Washington University. And with that, I'll take any questions.
Thanks. Beautiful talk. Very nice. I have a question. You showed in the beginning of in one of the beginning slides, you showed the correlation between the germinal center and TFH. There was a correlation plot, a scatter plot, where you showed that there's a high correlation. Yeah, this one. Can you explain this? So this is what? So this is TFH versus.
Yeah, so these are just TFH cells within the lymph nodes for these two individuals here. So the green would be green is donor 4, blue is donor 5. And this is just the frequency of those over time. And then this would be like a reciprocal of the D50 index, just as a measure of clonality. So an increase here would be more clonal. And so basically what we're seeing here is that the clonality correlates with the frequency of the TFH for each of those time points.
So why would that be surprising? Can you?
It's not, I don't think it's super surprising. It's good to confirm. I guess the alternative hypothesis is that you just get lots of unique clones that come in and make up the germinal center reaction. Unlikely, but it's just kind of nice to see how the expansion contraction correlates with it.
Yes. And there are a few outliers also, right? So it's not super consistent. Yeah.
Yeah. I mean, it is difficult to be super consistent in sampling these sites, right? You know, you could be off a centimeter and it could affect how many cells you recover and things like that. So
I see.
There's some nuance into the procedure.
So do you because you mentioned that, do you have replicates where you do twice the same thing? So do you find then consistent patterns, biological replicates, so 2 times the same sampling?
No, I don't think we did that too much. I don't think people like getting needles in their armpits too much.
But it would be nice to see if what we see is actually representative or not. Yeah. Because we know even from bulk sequencing, where you can get many more samples, if you do twice the same blood draw, even there you have low overlap. So yeah. Yeah. And then can you go to the network plot where you showed lymph? Yes, this one. So there is not so much overlap between lymph node and PBMC. Do I see that correctly? So there is more internal connections than cross connections? Is that? Yeah.
Yeah, that's true. I think one thing to keep in mind is that the cTFH, you know, fraction of just bulk PBMCs is quite low. We're talking about like 0.5% of CD4s maybe. And we didn't do any sort of targeted sorting of those cTFH to get good depth on those. So we're a little bit limited in terms of how deep we were able to sample the cTFH by sampling all the PBMCs.
Yeah, but at least the blue part shows that there's a strong interconnection across the time points that you sampled, basically. So there is strong conservation, which is quite nice, I guess. Yeah. Okay. And then about the antigen the TCR orphanization. So all of this data is not antigen specific, and for some of them you then try to find out what they are specific for.
Exactly. Yeah.
Okay. So is there future work where you have more antigen binding information then, except for if you?
Yeah, I mean, that was something we would love to do. To be frank, epitope discovery in flu hasn't progressed very much in quite some time. So there's not a lot of class 2 epitopes that we can use to sort of track these things like we did with the COVID study. And also the people in the COVID study were all naive to COVID, really. So you get these beautiful responses within those individuals, whereas in the flu vaccine, people have very different responses. Some respond very well, some don't respond at all. So the variability between peoples here is also kind of a confounding factor. But yeah, if we were able to identify, some more immunodominant epitopes that we could track out over time with these people, that'd be great.
Got it. Got it. Yeah. No, it's beautiful data. It's very nice. Are there more questions from the audience? Otherwise, thanks, Stefan, again, and thanks everyone for attending today. And we have another seminar in February, and it will be then more on antibodies and protein language models.